The next session that we've got is research that's been funded by ALK-positive, updates from researchers on the LUNGevity and GO-2 partnership research initiatives. And the format here is that all but a couple of the presentations are being done live, but remotely. Two of the presentations, they were not able to be here in real time. So I've pre-recorded on those, and we'll be playing a video on those. One's just a very short 2-minute introduction, and the other is a 10-minute 10-minute presentation from Dr. Bivona. It was actually a 14-minute presentation, and I sped it up. Before we get onto that, I think that everybody's aware, but just to be really clear, there's a mission for our organization, ALK-positive, and I haven't heard that mission mentioned yet. And it is central to everything that we do. And everything that we do has to somehow be able to relate to that mission. If it doesn't relate to that mission, we're not going to do it. And that research, the mission is very simple. The mission is to improve the life expectancy and quality of life for ALK-positive cancer patients worldwide. So there's a few things to notice there. There's no mention specifically of lungs, so it's ALK-positive cancer. It's worldwide, and it's not just focused on life expectancy, but also quality of life. Just skipping back to the presentation from Sam and Colin, they have done something really amazing that they didn't mention, way too modest, but they should have mentioned it. And so they mentioned that it was 3 awards of $250,000 each, total of $750,000. What Colin did not mention was that LCRF actually put in $150,000 of that $750,000. So thank you. Another thing that was not mentioned was the tireless amount of work that's done by reviewers in reviewing these. And these reviewers are not just the ALK-positive reviewers, and they certainly put in a ton of time, but also we've got many doctors and biostatisticians that are organized by LUNGevity and LCRF and GO-2 Foundation, and all the leading experts, the Dr. Cammages and Dr. Riley's, and surely the very best of the best, all give their time. And so if we could put our hands together to them for all the time that they spend, we could not do it without them. We've got some expertise on our internal reviewers now, some really amazing, but certainly 3 or 4 years ago when we were doing this, we really relied on the science side with the external scientists. So thank you very much. Also, Scott Schell mentioned that we've raised $7 million and that we're looking to get that greatly bigger numbers before too long. And a big part of that until now, truly everything we've raised has been by patients, patients' families, patient support crews, and you can keep on doing that. Spread the word, tell your stories. There is nothing more powerful than your stories. And every time you meet someone, they ask you, did you smoke? You tell them the story. You got diagnosed. Are you still working? You tell them your story. Just share your story. It's amazing how really the positive reaction that you get. So with that, we're going to have a presentation first from Dr. Upal Basu Roy. Dr. Upal Basu Roy is the Director of Research at LUNGevity. We did our first series of awards through LUNGevity in 2018, and we did a second series of awards through LUNGevity in 2020. And I'm going to say Upal, because everyone knows him as Upal. Upal has been absolutely central to everything that we've done. And Upal's a friend of mine. I can be sitting out on the lake paddling, sending him photos of the scenery, and he loves it. So I'll hand it over to Dr. Basu Roy. Upal, if you could share with us kind of what the review process is, that would be fantastic. Thank you.
First of all, Colin, before I get started, I just want to say it's such a pleasure to see you, my friend, and I wish I was sitting there with you, giving you a hug, and I wish I was sitting with all of you, but I'm there in spirit and waving at all of you all the way from New York City. So again, my name is Dr. Upal Basu Roy, and I am the Executive Director of Research at LUNGevity Foundation. And the foundation was started in 2001 by a group of 7 patients in Chicago, and LUNGevity was a very grassroots level organization. But one thing that LUNGevity always did right since its inception was it always funded research. So we are actually the oldest lung cancer patient advocacy group who've always been funding research, and research is central to our goal and to our mission. And we have 2 goals, which is to improve outcomes for patients diagnosed with lung cancer. And research is the cornerstone of improving outcomes, as we all know. And the second goal that we have is to improve how people are living with the disease. And again, going back to research, I want to take this moment to thank all of you for your partnership. Again, the partnership with the ALK-positive started in 2017 with a conversation, and all of you took a leap of faith with us. And now it's blossomed into 3 partnerships, as Colin mentioned. So thank you again for your partnership. Thank you again for trusting us to be stewards of both your scientific priorities and also your funds. And again, this is incredibly personal to me as well, because 3 weeks ago, a dear friend of mine called me up from India and said that her mom was diagnosed with ALK-positive lung cancer. So I can tell you right now that the fight is far from over, and we are in this together. So I wanted to spend a little bit of time telling you about how we decide on which projects to fund. And then you'll be hearing from some of the smartest, the kindest, and the most awesome scientists from the world with whom I have the privilege of working with, and who are here to tell you how they were changing, what research they're doing to make sure that outcomes of ALK-positive lung cancer is changed. So I will actually tell you a little bit about how we are selecting the 2022 awardees, and this process is the same for 2018 and 2020. Now, we typically started with something called requests for applications that went out January of 2022. And then we received 15 letters of intent in February. And letters of intent, look at them as the elevator pitch for a project. Now, the LOIs were reviewed by a panel of experts, and the experts are actually you, community members, who we call the research review panel. So we have members from the ALK-positive group reviewing these LOIs along with KOLs, along with scientists who specialize in ALK-positive research, and also with biostatisticians. So keep in mind that it's you who are deciding what research to fund. So it's very aligned with your priorities. Now, from the 15 LOIs that we received, we invited 7 scientists. And again, we received applications from all over the world. We invited 7 scientists to submit what we call a full application. Now, the full application is essentially the pitch deck where the scientists tell us again in more detail what that research is going to look like. And we received the full applications May of this year. And then we had what is called a study section. A study section is essentially where all the scientists, the reviewers, we all hunker down in the same room and go through the proposals and decide which are the most important from both a science perspective and also, I think even more importantly, from a patient perspective. And again, in the good old days, we would actually sit in a room and do that. But now, thanks to COVID, we actually do that across Zoom. And we finished the study section in June of 2022. And now the ALK-positive board is discussing the study section, and we will be announcing the winners shortly. Now, what is the secret sauce to the success of the ALK-positive LUNGevity partnership? The secret sauce is really a true collaboration between patients and researchers. And that's what makes this such a phenomenally important partnership. On the left side, what you see are patient reviewers, caregiver reviewers from your community, the research review panel. They spend a lot of time going through the proposals, going through the LOIs, again, making sure that the research is aligned with your priorities. And on the right side, you see are the researchers. And I think all of you know these researchers. In fact, a lot of them are your clinicians. So we have a discussion where these 2 groups are sitting together and deciding who will be funded. And I also wanted to share some numbers with you. What does that look like? Colin mentioned that reviewers spend a lot of time, but I wanted to put that in context. So we received 15 LOIs. That means 161 pages were reviewed by this group. And then we received 7 full applications. And that means 470 pages of research were reviewed by this group. And they spend around 670 hours reviewing these grants. So that should tell you that this is a very, very intense process. But again, very grateful to all of you for your support. But again, the secret sauce is you guys, because you will be choosing who the winners are. And this is the study section in action. Again, this could be sitting in a room, but these are the reviewers sitting in a Zoom room discussing the grants. And I just wanted to end my presentation just sort of putting in context what the research you funded has done for the ALK-positive treatment landscape. So this is the 2022 ALK-positive non-small cell lung cancer treatment landscape, and I think all of you are very, very, very familiar with it. And what I've highlighted in red boxes are drugs that are currently used as first-line treatment in the United States. Now, of course, the first-line treatment or the first treatment that a patient newly diagnosed with ALK-positive lung cancer is highly dependent on which country you live in as well, because certain drugs are approved in certain countries. But the red boxes specifically are those drugs, or I would say pills, targeted therapies that are prescribed as the first treatment in the United States. Now, this is what the landscape looks like now. Now, let's reflect a little bit further on. What will the landscape look like? And this is essentially the research grants that you funded. And I'm just going to introduce the awardees because they will be actually telling you what their research is about. But the reason I sort of bring this up is on the top, you see the 2018 awardees. Now, Dr. Awad and Dr. Chiarle, they're looking at characterization of ALK immunologic responses. And what you see in blue are the 2018 awardees. Now, the 3 2018 awardees are looking at a fundamentally important piece of information. How can we make immunotherapy work in ALK-positive lung cancer? Now, currently, the immunotherapies that are approved for lung cancer, for example, Keytruda, Opdivo, Imfinzi, we know that they do not work in ALK-positive lung cancer. Now, the question is, why do they not work? The question is, are there other pieces that can work, which is essentially and fundamentally what these 3 research teams are trying to investigate. Dr. Awad and Dr. Chiarle and Dr. Gainor, they're looking at a very interesting and a very, very important part of the immune system, which is adaptive immunity. And Dr. Nemenoff is looking at the complement system, which is a part of the innate immune response. Keep in mind that our immune system has 2 branches, the innate immune system and the adaptive immune system. And the reason I bring that up is these 3 researchers are taking a very comprehensive look at how the immune system can be mobilized to treat ALK-positive lung cancer. Now, on the bottom, you see the 2022 awardees. And again, I should mention that Dr. Awad and Dr. Chiarle are continuing the 2018 project, and the 2020 project is very specific to taking the information of the 2018 project and developing a vaccine for ALK-positive lung cancer. Now, let's look at the 2 other awardees, Dr. Dagogo-Jack and Dr. Frady and Dr. Bar-Peled. Dr. Dagogo-Jack is asking, again, a very, very important question, very relevant to the ALK-positive community. How can we use 2 pills instead of 1 to combat ALK-independent resistance, mechanisms of resistance to lorlatinib? As I mentioned, lorlatinib is a drug that is being used as the first treatment for ALK-positive lung cancer. And we also know that lorlatinib may stop working for some patients. And Dr. Dagogo-Jack is answering that question. What can we do when lorlatinib stops working? Can we add another pill to that drug? Now, Dr. Frady and Dr. Bar-Peled are not focusing on pills. They are actually looking at a very new type of drug development, which is protein degradation. And they're looking at developing drugs that can actually degrade or destroy the ALK protein. So again, a very different mechanism from the pills that we think of, or the TKIs or the tyrosine kinase inhibitors. So hopefully with this snapshot, I've convinced you that the research that you're funding and supporting is going to completely transform the ALK-positive treatment landscape. And with that, I'll again end with a word of gratitude to all of you for fostering an ecosystem of research. And I think that's incredibly important. And I'm going to go back here again and remind you why that's important. Your support has fostered a collaboration between Dr. Awad and Dr. Chiarle. Your support has fostered a collaboration between Dr. Freti and Dr. Bar-Peled, and we cannot underestimate the importance of collaboration in driving research. And again, I'll end here by reminding all of you that we are in this together. And I do want to point out the photo on the left. It is very near and dear to my heart. In fact, this was the last photo we patient advocates took in San Diego in 2020 before the pandemic hit. And again, these are some of my favorite people in the advocacy community. I think you recognize Colin, obviously you recognize Dr. Amy Moore, Janet Freeman-Daly, you obviously recognize Dr. Christine Lovely, and then of course, Jill Feldman and Marcia Horn. Again, we are in this together, and thank you again for your partnership.
Thank you, Upal. That was a terrific overview. We're going to have presentations by all of the longevity researchers, and then we're going to have a brief presentation by Dr. King, who's the Director of Research at the GO-2 Foundation. And then after that, we'll have the presentations by Dr. Lovely and Dr. Bivona, who are doing a collaborative project through GO-2 Foundation for ALK-positive. So we're going to start off with Dr. Roberto Chiarle presenting about the 2018 award that was given to Dr. Awad.
Hello, hi Colin. Can you hear me?
We can hear you terrific, thank you.
Great, yeah, it's fantastic to be here. This is starting to begin a very important summer appointment for everybody of us. So it's great that you keep organizing, and the ALK-positive group is so exceptional in doing this type of events. So let's start my presentation without any further ado, because I think we need to be on time. I'm going to share my screen and do a virtual live presentation. Okay, I hope everybody can see my screen. Okay, so I'm here to present a project that has been developed together with Dr. Awad. He couldn't be here this time, but I'm sure he will be next. And the presentation is about an award won in 2018, as Upal summarized. That was the first seed to understand how immunological responses against ALK are developed in patients. And then the next presentation later will be how we will exploit this understanding into building something more translational and a phase 1 trial for an ALK vaccine. So in this presentation, in this 8-minute presentation, I will discuss about 2 topics. We decided to investigate how patients react to the presence of the ALK-positive lung cancer by investigating 2 types of immune responses. One is an antibody-mediated immune response, and the second is a T-cell mediated immune response. And they are different because, of course, everybody of you know what antibody are and what T-cell responses are. They are like 2 sides of the immune system that reacts against a foreign protein. In this case is ALK. ALK can be considered a new antigen in this type of tumor because ALK is normally expressed not really in normal cells, but only in tumor cells after a translocation and a gene rearrangement happens. And now the protein is expressed very high level in the tumors. I will comment on this more later in the next presentation about the ALK vaccine. So we imagine that when the ALK protein is expressed by tumor cells, the immune system would react against this protein and generate a specific immunity that can be measured eventually by antibody responses, by T-cell responses. So to test antibody responses, we collected, of course, together with Dr. Awad, more than 100 serum. You can do that on the peripheral blood or on the serum. And then even with a little amount, you can test whether there are in the serum of patients specific antibodies against ALK. And because there was, and there is yet not a commercial test to do so, it's not like COVID where we have a commercial test that can be easily used to detect antibody against COVID. In the case of ALK, it's not existing. Maybe it will in the future as a readout of the anti-ALK immune responses. But we needed to do in the lab. And to develop this assay, we basically incubated the sera of the patient against recombinant protein of ALK. And there are here, this is a methodology called Western blot, by which different type of ALK fusion protein, you can see we have one type of fusion called NPM-ALK, another type of fusion called ML4-ALK, and the third type of fusion called full length, which is not the fusion, actually is the entire transmembrane receptor. And you can incubate this recombinant protein with the sera of the patient and see if antibody recognize this protein and can be detected. And to make the story short, here in the figure on the right, you can see that if you test the sera from an ALK negative lung cancer patient, so a patient that has a different type of lung cancer, not ALK, you see that basically the membrane is completely white. In contrast, if you test this membrane with sera from lung cancer patients that are ALK positive, now you see all these bands that testify that there is a reactivity, so a presence of antibody against different type of ALK proteins. And here on the left, you can see the graphical summarization of all this data. You see that the vast majority of the patients develop very weak immune responses, but there are about 20 to 25 patients are these blue dots, even very, very high, that develop very strong and very intense immune responses in terms of antibody generation against ALK. So what does this mean practically? Well, we are trying to understand what is the implication. And so first we decided to map where this antibody bind in the ALK protein. And we did an array of different ALK peptides that cover basically the entire protein that is fused in ALK rearranged lung cancer patients. And you see that the antibody basically mapped to two regions. One is the very early initial part of the protein, and then the second region is the late, or we call also C-terminal or T domain of the protein. And why the antibody bind to these two regions, we don't know exactly, probably because these two regions are more diverse and they are more different compared to other tyrosine kinases that we have in the body, like ROS1 or RET, that have similarity to ALK. And next we ask whether the presence of this antibody could be associated with a better prognosis. Of course, technically speaking, this antibody, they are not supposed to do anything against the tumor cells because the ALK protein in lung cancer is internal inside the cells. So it's not exposed on the surface of the tumor cells. So even if there are circulating antibody, they technically cannot bind to tumor cells. However, they could be a readout or an indirect measurements that there is some immune response in patient against ALK, and this could be beneficial to extend survival. And so in this analysis in which we tested more than 100 cases, you can see that patients that develop spontaneously very high antibody level, they tended to have a better survival than patients who did not develop this type of antibodies. So this could imply that if there is a spontaneous immune response against ALK that we see in about 20 to 25% of the patient, this could be associated to an extended survival. Next, we decided to characterize the T-cell responses. So the T-cells are lymphocytes that generate a direct killing activity against tumor cells. So technically speaking, this type of immune response would be more associated with an anti-tumor activity. However, it's very complicated to test T-cell responses unless you do not know, or you know the antigen of ALK that is recognized by T lymphocytes. So to understand what type of T-cell responses are developed by patients with ALK-positive lung cancer, we first wanted to characterize the immune system of these tumors. And we stained for this molecule that is called MHC class 1. This is a very important molecule that is expressed by tumor cells and also by non-tumoral cells, of course. And this protein or this complex mediates the immune response of T cells, in particular CD8 cytotoxic T cells. And by doing this analysis, we could demonstrate that basically 100% of tumor cells that express ALK, you see here this brown cells are ALK-positive cells. They also express this molecule that is important to engage T cells and generate immune responses. But then we needed to characterize what were the part of the protein of ALK recognized by these T lymphocytes. And this was a long journey done in collaboration with Cathy Yu, Alice Reynolds at the Dana-Farber Cancer Institute and at the Mass Spectrometry Institute at the Broad. Through this journey, we identify few peptides, actually 4 so far, that are generated by tumor cells from ALK. So it's ALK that is fragmented into individual peptide, and this peptide are presented on the surface of the tumor cells. And this is very important because we need to know this peptide to detect the T cell responses. In an absence of ALK peptides, it will be extremely difficult to be sure that the T cell responses that we measure are really ALK specific and not directed against different type of antigens. So we first identify these peptides, and I will comment more in the presentation about the ALK vaccine. And by using this peptide, we try to characterize the immune response. Here is an example of this peptide directly on a cell line that is an ALK-positive cell line, and we were able to characterize the presence of ALK peptide even directly on tumor biopsy from patients, implying that the tumor cells in the lung, they do present ALK on the surface, and so they can be detected by T lymphocytes. And so next we develop a specific assay to detect T cell responses against ALK. This is a rather complicated slide, but to make the story relatively short, or the assay relatively simple to understand, we need to have some cells that present, this is the terminology used in the immunology field, so we need cells that present the ALK peptide or the ALK antigen to the T cells. And these presenting cells can be dendritic cells, can be peripheral blood in general, or can be B cells. We use all different type of presenting cells, we stimulate them, we incubate with the ALK peptide, and then we went on this presentation system with the T cells directly from patient. And here I can give you one example. This is called ELISpot, and in this assay, every single dot that you see here, this black dot, they represent one lymphocyte or a group of lymphocytes that recognize the ALK peptide in this case. So for example, from this patient, the CD8 T cells, they recognize there were a lot of cells, you see how many dots there are in these cells, that recognize this particular ALK peptide that we discovered through mass spectrometry. And so by this assay, we were able to establish that, again, about 10 to 15%, very similar to the antibody assay that I showed before, about 10 to 15% of the ALK-positive lung cancer patient, they spontaneously have this type of T cell responses against this ALK peptide. So this was very important for us to understand that ALK is really seen as a, and I'm concluding the presentation with these acknowledgements of the people that work. So ALK is really seen as an antigen by the immune system. So like a new molecule or a different molecule that the immune system can target, and we can exploit this mechanism to develop a vaccine. This will be the topic of the next presentation that will be done soon. Thank you very much.
That was outstanding. Thank you. One of the greatest challenges we've got here is that we've got a level of scientific understanding that can range from having no scientific understanding whatsoever and probably not even knowing what a cell is, all the way through to our professor of cell and gene therapy at the other end of the spectrum. So trying to get presentations that is understood by everyone can be a bit of a challenge. This is all going to be recorded, and I do encourage you to watch it as many times as you need to understand it, which might be 2 or 3 or 4 times. I know when I was reviewing proposals, very often I had to read proposals 4 or 5 times before I knew what it was all about. We have next, is it Dr. Gainor or Dr. Nemenoff? Dr. Nemenoff. So if you're there, Dr. Nemenoff, we are ready for you to go live.
Okay, can everybody hear me? Is that okay?
Perfect.
Good. Let me share my screen. So again, it's always wonderful to to talk to this group. It always makes you sort of puts things in perspective as to what you're doing. And I'm going to give you an update on a project that was funded in 2018 by, as Upal mentioned, and a project that we're still going on, and we're still pushing ahead with other funding. And a project, warts and all, I'm going to present some difficulties that we've had from the translation part of it. And I'm also going to talk about some work that has spun off from this related to ALK-positive lung cancer, which we've started in the last year and hope to relate to this. So the project that we were funded on was targeting the complement pathway in ALK-positive lung cancer. And the complement pathway is usually one of those pathways, even in a group of scientists, that tends to put people to sleep because it's an incredibly complicated pathway. And I'm really not going to go through all of it for this group, but it is, as Upal mentioned, it is a part of the innate immune system that generally is associated with a very rapid response to pathogens and is a very ancient part of the innate immune system. But importantly, it's also an interface between innate immunity, and you heard about T cells, I'm going to come back to some of that, and adaptive immunity. And so it is a regulator of this crosstalk between different parts of the immune system. Complement seemed an attractive target to look at in terms of cancer because complement activation can lead to direct killing of target cells. And so ideally, perhaps if we could activate complement, we can kill cancer cells. But complement activation has also been associated with chronic inflammation and therefore can promote cancer. So it may be a double-edged sword, and that's one of the complexities in this field and continues to be one of the complexities. One of the things that drew us into this was that complement inhibitors at that time have been approved for a variety of inflammatory diseases, and that continues to advance. I'm going to touch on that as well. And so it appeared that one of the strengths of this project was to be able to develop new therapies and new types of drugs to improve the response in ALK-positive lung cancer patients. So this is sort of a schematic of the complement pathway. It involves a series of very complex events associated with tagging specific cells, and it results in the production of these inflammatory mediators. And this is the part of the complement system that is associated with killing cells. So I'm not going to really dwell on that because it generally is a very complex system. So this is a brief summary of the studies that were supported starting in 2018 by ALK-positive. So we were able to use a mouse model, which is an immunocompetent mouse model that's highly relevant to human tumors, a model of ALK-positive lung cancer. And we were able to show that complement inhibitors in this mouse model were effective in blocking lung cancer progression. So down here, we have two different models. These are cells which we implant ALK-positive cells directly into mice, and we can follow tumor growth. And you can see this is a tumor growth, and this is using either pharmacological or genetic inhibitors of the complement pathway. And in both cases, you get very strong tumor shrinkage and inhibition. So that was pretty exciting to us. We also have more recently shown that we can target regulatory proteins which block the negative effects on cancer cells. And so that may represent an additional therapeutic approach, and that's still work under investigation. These are publications that came out of these studies, and we were fortunate enough to acquire other funding and are incredibly grateful to LUNGevity and the ALK-positive for starting this project. And we're still going on, and we're still going forward with this. However, a major obstacle, and this is something that I've talked about with Colin, with other people, although several complement inhibitors have been developed that are either approved or in clinical trials, it has been very difficult to engage companies that have this, or even companies that are startups that are developing drugs not approved yet, to get involved in these cancer trials. So there are antibodies against C5. This is a drug called eculizumab, which has been around a long time, and it's approved for the FDA for these inflammatory diseases, paroxysmal nocturnal hemoglobinuria and myasthenia gravis. These agents are very well tolerated. So one of the appeals when we started this project was if we could develop a therapeutic approach, these drugs can actually be given to cancer patients over long periods of time and don't have a lot of toxicity. There are other specific inhibitors which are currently trying to be developed for clinical trials. And there's more recently an FDA-approved new complement inhibitor which directly inhibits C3. And so the landscape is rich in potential agents, But the problem has been that even to this time, there are very few trials in all kinds of cancer using complement inhibitors. We've had conversations with numerous pharmaceutical companies, and there seems to be a reluctance to move into this space. And I've had patients reach out to me, patient advocates, to see if we have updates, and we're still continuing these conversations. We have a ongoing discussion with a startup company in Boston that is developing a fusion complement inhibitor, but this is still a long way away, and they're only starting phase 1 studies, and their initial targets are not cancer. So this has really been an obstacle in terms of moving forward on the clinical side, and we continue to look down this pathway. There was a clinical trial in Colorado that was initiated by a Chinese pharmaceutical company. I checked last week, and this has not enrolled any patients here. So this is an obstacle. And as a basic scientist, we don't have a lot of leverage. Ross Cameron has helped us with some conversations, but unfortunately, this has not been successful. So in the last couple of minutes, I want to sort of tell you about a new project that we're starting, which we're going to interface with the complement project, but is really related to trying to get a better understanding of how the immune system works in terms of ALK-positive lung cancer. So this is what is known as a waterfall plot, and just shows the response of individual patients to, in this case, alectinib. And this is lifted from a paper of quite a while ago. And what you can see is that some patients show a very strong response, and some patients show a more modest response, and some people don't show any response. So what underlies the inherent differences in this response? And is there something inherently different in the cancer cells? That's a possibility, but we're interested in potentially, are there differences in the way the cancer cells interact with the tumor microenvironment? And in particular, can we model this in mice? There are numerous studies that have used human samples to try to develop correlations, but those really can only develop correlations. In preclinical mouse models, we can potentially identify the molecules and the pathways that mediate this difference in response, and this would lead ultimately to the development of new therapies in combination with FDA-approved therapies. So I'm going to show you just a little bit of data from that project. So in light of that, we have developed a panel of murine ALK-positive lung cancer cell lines, and these are developed using technology, CRISPR technology. The initial one was developed by Andrea Ventura at Memorial Sloan Kettering. We've developed another 2 or 3, and if we take these cells and we look at their response to a ALK-specific inhibitor such as alectinib, you can see they all look pretty similar. This is just how well the cells respond to ALK inhibition. So in the dish, in vitro, these cells are all the same. And the differences, if there are going to be differences in vivo, they're not going to be due to the properties of the cells in vitro. So we've now done this experiment where we've now implanted each of these cells into immunocompetent mice shown up here. And what you can see is that there's really quite a different response. So the EA2 cells show a complete response clinically. Basically, the tumors shrink, they go away. If we stop treatment, the tumors never come back. And it seems that these would represent a complete response in a patient. On the other hand, both these EA1 and the EA3 system do not show residual disease. So EA1, you see tumor shrinkage, but we can detect using micro-CT technology still the presence of residual tumors, similar to what you would see in patients. And the minute we stop the treatment, these tumors roar right back. And this is similar for these EA3 cells. So this represents at least a beginning to look at a complete response versus a partial response in these patients. To test the role of the immune system, we took these same cell lines and we implanted them into mice that don't have an immune system. So the question here is, does the adaptive immune system, that is T cells and B cells, play a role in the clinical responses to alectinib in this case? And what you can see that in all 3 of these cell lines, if we treat them with alectinib and continue the treatment, while we see initial tumor shrinkage, very rapidly these cells reprogram and they grow right back. So our take home, and this was really a big surprise to us, that in a case where the adaptive immune system is not there, the responses are very different, saying that even though conventional immunotherapy such as checkpoint inhibitors don't work in these patients, the adaptive immune system must be critical in maintaining the responses to alectinib and somehow allows you to have longer and better responses. We've begun to look at these tumors, and this is just one slide showing. So I'm comparing here, these are the EA1 tumors that basically have a a partial response, residual disease, and will grow back if you stop treatment. And the red stains here, we're just staining these tumor sections for T cells. And you could, in this case, CD8 T cells. And you can see that there are a few of these. But if you look at the cells that show a complete response in this model, you see many more T cells at baseline and even a further increase when we treat these cells for alectinib for a few days. So this says, at least in this model, that the presence of T cells in the tumor at baseline and the ability of TKIs to increase T cells may play a role in the duration and the depth of response. So just to summarize all these studies, so in multiple cancers, including ALK-positive lung cancer, activation of complement regulates anti-tumor immunity. Complement inhibitors have been approved for several rare diseases and are being tested in other settings, including COVID and other cancers, but not very actively in cancer. And one of the things we hope to continue to do is to try to develop novel inhibitors and work with pharma or developmental companies to test new agents to see if they will be effective in these models. ALK-positive lung cancer is a low response rate to anti-checkpoint inhibitors. However, the role of the immune system in these tumors is not well understood. In the previous talk, you can clearly see that this is an active area of investigation by many labs. We believe that T cells play a critical role in determining the clinical response, that is the time to progression to TKIs in ALK-positive lung cancer. And we hope to understand why there are different T cells in different tumors and how we can harness approaches to increase T cell infiltration in these tumors to develop new combination therapies. So with that, I just want to thank all the people who've done the work. We've been fortunate enough, after obtaining funding from LUNGevity and the ALK-positives, to obtain money from the DoD and the NIH. And also I want to thank the Thoracic Oncology Research Initiative at the University of Colorado, which has been instrumental in developing these models, and hope to work with them in trying to develop these new therapeutic approaches. And I'll close with that.
Thank you very much, Dr. Nemenoff. So I'm going to have everyone stand for a minute or two. Stretch your legs, spread your arms out, wave around. So in case you didn't quite get it from that presentation, the funds that we invested in research with Dr. Nemenoff in 2018 not only led to some really good research that's still ongoing, but the research that he did for that then led to him getting significant funding from Department of Defense, and I'm talking seven-figure number as a result. So this is leveraging of our donations. It's not just what gets done with our research, but the fact that that can lead to other research that they get paid from other grants. Lastly, we have Dr. Gainor for the 2018 awards. So if you're there, Dr. Gainor, it's all yours.
Great. Thanks so much. Can you see my slides okay?
Yes, we can. Thank you.
Perfect. Hi, everyone.
And sorry, before you get started, I'm sorry, but everything's been running a little bit on overtime. So we'll ask you to keep to time would be fantastic. Thank you.
Yes, I'll certainly do that. So thank you so much for having me. I think this is a really inspiring event. And I also think that this award is particularly meaningful because it comes from our patient community. And I would echo the sentiments from one of the prior sessions, the comment made by Satoshi Yoda, who is a postdoc in our group, just echoing how hard everyone is working to really try to make progress as rapid as possible. So my talk today is really going to be focused on our work to develop novel immunotherapies for ALK-positive lung cancer. And
Dr. Gainor, I'm sorry to interrupt. Our AV person, Keith, has just mentioned to me that somehow your slides are coming across in the presentation mode rather than the viewer mode.
Is that better?
He says it's fine now.
Okay. So as you've just heard from really the last three talks, including my own, we're all talking about immunotherapy. And I think one of the reasons why we're trying to develop immunotherapies is if we think about the aspects of the immune system that can be attractive as anti-cancer therapeutic is it can be exquisitely specific, but most importantly, it has this potential for memory. And in the clinic, we've seen that immunotherapies are really the thing that is producing what we dare say cures in the stage 4 setting. So I think this is why we're working so hard to try to identify novel forms of immunotherapy to target ALK. Now, what you've already heard is that, immunotherapy, the term itself is quite broad. So it actually captures multiple different forms of therapy. So we've heard about checkpoint inhibitors or PD-1 inhibitors. We've heard about vaccines. And then I'm going to talk a little bit about cellular therapy at the end of my talk. So I'm going to start with checkpoint inhibitors because checkpoint inhibitors, no doubt, these are already approved in non-small cell lung cancer. And as you just heard from Dr. Nemenoff, these agents have, while they can be very active for some patients, what we've seen is that they haven't produced high response rates in patients without positive lung cancer. Now, much of our work was focused on trying to understand, why ALK-positive tumors are less responsive. And one thing to highlight is that determining responsiveness to immunotherapies, is actually quite complicated because not only do you have to look at the tumor itself, but also the microenvironment, that is the neighborhood that surrounds it, because it's not just all tumor cells. There's actually a complex microenvironment around it that includes support structures, potentially other immune cells, and these may be playing a role in dictating response. And so one of the ways that we wanted to approach this was recognizing that not every patient can travel to Mass General to participate in our research studies, is we actually wanted to develop a web-based platform that could allow anyone, in the United States. The goal is to make this global, but for regulatory reasons, we had to start US-based. But could we create a web-based platform that enables any patient to participate in this form of research? And this is really translational work. This is based on medical records, but also tissue-based specimens. And can we integrate genomic data with immune profiling and clinical data to identify markers of response? And that led to this, it's called the Enigma Plus protocol. This is elucidating novel immune and genomic markers for ALK. This is something that I'm doing with Jessica Lin is leading this project, but this is in conjunction with the GO-2 Foundation, where any patient can actually go to this website and get a call and actually sign up for this protocol. And then our research coordinators will actually request tissue and actually do some of the work that you're going to hear about. So what we're doing is really focusing on two different, so genomic markers, so that's doing broad sequencing. And then what I'm showing here, this is multiplexed immunofluorescence. So this is a way to try to, on a single slide, actually look at multiple markers of the immune system. So up to 6 markers in a single slide. And one of the nice aspects of that is that it actually allows us to take one slide and look at 6 things. And what we've learned is that actually that tumors that are more responsive to the checkpoint inhibitors actually have these immunity hubs, that the immune system isn't just this homogeneous area, that there are these discrete hubs. You can see in yellow here, this is a specific marker looking at certain secreted things from immune cells. And so we see that there are actually these discrete hubs. So there's this spatial orientation. Furthermore, there are actually good hubs and bad hubs. And we find that if you have more of these good hubs, that you're much more likely to respond to checkpoint inhibitor. And so a big part of our work right now is how can we actually induce these hubs? Because what we find is that in ALK-positive lung cancer, they lack those hubs. These tumors lack those hubs. So what can we do to actually induce them? And so we're doing work in the lab now to see what other therapies we can add to try to induce these hubs, because we think this is actually where the immune response is being initiated. In the last couple of minutes, I wanted to focus on a second aspect of this project, which is cellular therapy. For those of you unfamiliar with cellular therapy, what this involves is actually taking immune cells out of the body, usually through just a simple phlebotomy procedure. So taking the immune cells out, genetically modifying them to basically target something more specifically. So you're giving every immune cell that you're removing the ability to now recognize something. And so what we're trying to do is develop ALK-specific immune cells. These are known as ALK-specific TCRs, or T cell receptors. And so the goal then is to then give these cells back to patients and use these now modified cells to be able to attack the cancer cells. And so this is very similar to what Dr. Chiarle was describing, is in order to actually develop these cells, in order to actually genetically modify them, first you actually need to find naturally occurring T cells against ALK. So in a way very similar to what he described, and in fact, we're working with Dr. Chiarle, and you'll hear at the end, we just recently received a 5-year grant to work on this together. But basically, can we start with identifying ALK-reactive T cells? And the short answer, because I know I'm at time, is yes. So on this slide, so every purple area, except for that last line, so each column represents a patient, and each increasing degree of purple, that represents us finding a T cell that's reacting against an ALK peptide. And so what we're doing right now is actually seeing how specific these can be, and actually then cloning them to identify the most specific and the strongest receptors against ALK, with the hopes of one day making this a therapeutic. So I know I'm at time. I did want to say that many people have supported this work. Thank you so much to the ALK-positive community. This actually is now going to continue as part of an NIH SPORE project, which is a 5-year collaborative grant, and that is being co-led by myself, Dr. Awad, and Dr. Chiarle are the co-leaders of that 5-year project. I also want to thank all our other funding sources, including the Venanzi family. So with that, I'm happy to take any questions.
I think we'll probably hold questions, and if we have time at the end, we'll bring those to the fore. If not, everyone that's remote or even here, feel free to, well, actually it's a bit hard for the people here, but I was going to say on the chat, you can put your questions into chat, and we will get those questions answered. Not necessarily today, but they'll get answered, and we'll figure out a way to get it back to the community. So thank you very much, Dr. Gainor. That was a great presentation. So I think next we have Dr. Chiarle again, is it? Terrific. So Dr. Chiarle, if you'd like to talk about the 2020 award, which is actually a phase 1 clinical trial, and update everyone. I know everyone in the community is extremely anxious to know where that's all up to. And so you have the floor again. Thank you.
Share my slides. Okay. So yes, this is the update about the phase 1 ALK vaccine clinical trial that again we've been implementing with Dr. Awad, and was graciously funded by the 2020 ALK-positive grant. Okay, so I've previously presented the key points of the anti-ALK immunity that we have basically discovered, identified by antibodies and T-cell responses. And this is just a visual image to show where ALK-positive cells stand in contrast to ALK-negative. So you can see, or can imagine at least, that the immune system can recognize this protein to be different from any other cells in the body. And the key point to develop an ALK vaccine is that we want the vaccine to be specific against the tumor cells. And so we need this key point that the tumor cells should be selectively and highly expressing the protein, in this case ALK. And this is true and confirmed. 100% of the tumor cells, as you know, they do express ALK. So the protein is present everywhere. And more importantly, the tumor cells are strongly addicted to ALK, and that's the reason why the ALK inhibitor works so well, because basically all the tumor cells depend on ALK to survive and proliferate. And finally, as I showed in the presentation before, ALK is immunogenic in patients spontaneously. And so there is a chance that by implementing or increasing this immunogenicity via vaccination, we could achieve stronger and better anti-tumor responses. So what do we need? What are the ingredients that we need to do a phase 1 trial? There are at least 4. I'm just showing the 4 more important. So we need to be sure that the tumor cells express all the components of the immune system that can be exploited via vaccination. And I previously showed the expression of class 1, and I will go briefly there. Then we need to identify the ALK immunogenic peptides, and I presented in the previous presentation a few examples of that. Then we need to demonstrate that this peptide, they really elicit an immune response. And then finally, probably most importantly, we need to show that this type of immune response can be translated into a therapeutic or anti-tumor effect. So to go briefly to these 4 ingredients to develop a phase 1 trial, so we show that basically 100% of patients with an ALK-positive lung cancer, they do express the machinery to present the antigen to the immune system. So this is important. We identify the ALK presented peptide by tumor cells. I show a little bit more details in the previous presentation. We identified 4 so far, and this identification was done in multiple cell line and even directly in tumor biopsy from patients, so testifying that the tumors are really presenting and showing this ALK peptide on their surface. And we need to show that there is spontaneous immunogenicity. Again, this was part of the previous presentation, but more importantly, we need to show that this peptide, these ALK peptides, are really immunogenic in a vaccination model. And so that's where the new data basically come. And we vaccinated mice that have the same class 1. These are weird names for non-immunologists, but this is the way class 1 molecules are labeled in the immune field. And so there are transgenic mice that express this human class 1 molecule, so they mimic exactly the immune responses that are seen in human patients. And you can vaccinate these mice with the peptides that we identify, and you can see by these bars that every single mouse, some very strong, some other a little bit weaker, but 100% of the mice responded by developing a specific immune response against the wanted ALK peptide. And we did this assay with all the ALK peptides, and all the ALK peptides work, and all 4 elicit specific anti-ALK immune responses. So we clearly demonstrate that these ALK peptides are immunogenic in preclinical model, which are transgenic mice. And finally, even probably more importantly, we need to demonstrate that this vaccination has an anti-tumor activity, because that's what we want to be sure before we start the clinical trial. And to do this, of course, we needed to develop a complex mouse model, immunocompetent, so with the immune system that is belonging to the mouse, but can recognize still the ALK peptide. I'm going fast through this part, but the key point is that we identify specific ALK peptides in the mouse model. This is one example. This is peptide number 7. And we team up with a group at MIT that developed a new modality of generating very strong anti-immune response against peptidic antigens by adding these amphiphilic tails. By the way, they are using this technology also for COVID, just for sake of knowledge. And by conjugating this peptide with this tail, we could elicit a very strong and specific against this precise ALK peptide immune response in the peripheral blood of mice. So you see that in non-vaccinated mice, there are basically no lymphocytes that recognize this peptide. After vaccination, 1 out of 5, 20% is a very high number in the peripheral blood because we have millions and millions of lymphocytes. So we are talking about millions of T cells that can recognize specifically this peptide. And you can see this is the same as I showed before, each of these blue dot is a cell of a group of cells, and you see how many of these cells react specifically against the ALK peptide we can elicit after vaccination. So this implies that a vaccination strategy with one single peptide is extremely potent to generate an ALK-specific immune response. Is this response therapeutic efficacious? Well, we needed to develop in mice, of course, preclinically, and we will use this data to present to the FDA when we'll submit to the FDA the approval to start a phase 1 clinical trial. So we needed basically to mimic how we plan to do a clinical trial in patient to mimic that in mice. So we have the tumor cells, and then we will start to vaccinate before or after the starting of an ALK inhibitor. In this case, it's lorlatinib, and we might add, yes or no, an immune checkpoint therapy. The immune checkpoint therapy don't really work against ALK-positive lung cancer. That's the previous discussion that we had. However, they might be beneficial to potentiate the effect of the vaccination. So despite they won't act against the tumor, they could further expand the immune responses during vaccination. So that's why they could be a reason to add immune checkpoint therapy during vaccination. And this is the summary of few years of work in multiple repeated experiments, which confirm all the time the same results. And you can see this dark line, whether you don't treat the mice or you treat only with ALK inhibitor immune check therapy, you can a little bit extend the survival of mice. However, all of them will eventually die. In contrast, when you start to add a vaccination, you can further delay the relapse of the tumor and so further extend the survival of mice. But more importantly, you can achieve a plateau. That's an important sign in the anti-cancer immune field, because when you reach a plateau, it means that you finally eradicate tumor cells and cure the mice in this case, and there is hope that we'll be able to cure patients. And in the best condition, which is this in combination with vaccination, lorlatinib, that you know is of course is an ALK TKI, and anti-CTLA-4, we could cure up to 70 to 80% of mice, which is impressive because it's really a big difference in contrast to 0% of cure with an ALK inhibitor alone. And what the vaccine is doing, of course, is making or transforming a tumor that is technically a little bit cold, so does not have a lot of immune response. And you remember the presentation by Dr. Nemenoff that showed that the tumors that do better, they have a little bit more of immune response from the beginning and after ALK TKI treatment. And what the vaccine does is really bringing into the tumor hundreds of thousands of cells in each single tumor. So you see all these dark cells are T lymphocytes that recognize the tumor, kill the tumor cells, and the tumor are much bigger and eventually eradicated. And the most important part, and I'm concluding basically the presentation, the most important part is that the vaccine is also very effective in preventing or eradicating metastatic lung cancer, in particular to the brain. So you see here on the left how in this mouse model that ALK-positive tumor cells that tend to go to the brain in the meningeal space, which is also seen in patients. However, when you combine the vaccination, the meningeal space is completely clean, and we can basically cure 100% or prevent 100%. You see all these 0, 0, 0. These are the three conditions with the vaccine. So we can completely prevent the metastatic disease or cure the metastatic disease in mice. So this is very important because, of course, metastasis to the brain is an important issue in patients. And so we check all the 4 boxes, and we are moving now implemented a phase 1 clinical trial in collaboration with Felicio Therapeutics, and the trial will be basically either vaccination alone or vaccination together with immune checkpoint therapy, as discussed before. And I stop here, and of course I thank ALK-positive and LUNGevity for all the support. ALK-positive has been tremendously important to move this project forward. Thank you very much.
That was a terrific presentation. So Dr. Awad and Dr. Chiarle's clinical trial is delayed, and he's doing more prep work, as you've seen, but it is definitely moving forwards, and it will get there. So we hope that once it does get to clinical trial, that all that groundwork makes it be as successful as it looks like it's going to be. We have 2 more presentations from the LUNGevity Awards. So we've got Dr. Dagogo-Jack, combination trial with the SHP2 inhibitor, and then Dr. Ifradi's presentation will be done by his partner, Dr. Liron Bar-Peled. So we'll have those 2 come on next. If everyone wants a bit of a stretch, now's a good time.
Okay, thank you, everyone. So we're going to pivot a little bit up from talking about the microenvironment, which I think is very important. Actually, I think that's the future of our therapies, but the purpose of this particular project was to try to think about things that might be happening in our tumors and growth signals that might be directly activating the tumors and causing what we call ALK-independent resistance or bypass signaling. And so I will explain how we came up with this project. And again, I think even before launching into the explanation, I want to thank you all for your support throughout all of this. And a lot of this work, a lot of the hypotheses that drive this work are really patient driven, in that it comes from patient derived biopsies and cell line models that helped inform which particular combinations to prioritize in our analyses. And so I think it's important to start with the central premise or hypothesis or the background. And most of you in the audience know by this point, based on talks and based on talks throughout the year and also talks you've heard today in other forums, that while we have improved our targeted therapies for ALK-positive lung cancer by really prioritizing next generation ALK inhibitors like alectinib or lorlatinib in the first and kind of second line setting, what we have started to observe is that there's an increasing prevalence of what we call ALK-independent resistance. And so the majority of tumors will eventually develop ALK-independent resistance such that they don't respond to further treatment with ALK monotherapy, ALK TKI monotherapy. When we try to study what drives ALK-independent resistance, this is really informed by studying or analyzing tissue specimens, but also largely informed by studying patient-derived models, so cell lines or mouse models, and doing functional analyses. There's been a variety of bypass pathways that have been identified. And this suggests that there may not just be one key bypass pathway that we need to target, but that there would be multiple. I think the encouraging thing is that in preclinical studies that we found that, if you add to a backbone of an ALK inhibitor, a drug targeting the most relevant bypass pathway in a given or individual tumor, that we are able to resensitize these cell lines to therapy, or we're able to stop the growth of the cell lines. And so that informed the hypothesis of this project. We wanted to develop clinical combinations that were targeting the most recurrently activated, the most, we thought, relevant bypass pathways based on our studies up to that point. And so this is just an illustration of a potential kind of clinical path or clinical journey. I assume that most people are starting with treatment with alectinib, but of course, this isn't me recommending alectinib over anything else. There's also brigatinib or lorlatinib. But just to really illustrate that after alectinib, we really see 2 phenotypes of resistance. So there's ALK-dependent and ALK-independent resistance. And for ALK-dependent resistance, there are targeted and personalized options, right? We have lorlatinib, we have TPX, we have the nuvalent drug in clinical trials. But for ALK-independent resistance, we don't really have a lot of personalized or targeted approaches, and all roads ultimately lead to chemotherapy. And so the goal of this was really to explore potential combinations that could be launched in that setting. And so to inform which combinations to pursue, we took it kind of back to the tumor, right? So we studied tumor biopsies, both plasma and tissue, at resistance to second generation ALK inhibitors. So alectinib, brigatinib, ceritinib, drugs like that, and lorlatinib, to really characterize what is the underlying driver of resistance in I guess, across tumors. And so these are pie charts summarizing that. What we saw and what others have described kind of before this, and you may be familiar with, is that in tumors that are resistant to second generation ALK inhibitors, and this is largely per the old paradigm where people receive crizotinib first and then receive the second generation ALK inhibitor. But we imagine that, similar themes may be true in the current context where you start with alectinib. We found that the majority of tumors are actually developing resistance at the hands of what we call ALK-dependent resistance. So it's acquired or new ALK point mutations. As mentioned, these are cases where we already have existing therapies that can be pursued. For the remaining tumors, we employed a variety of assays to try to tease out what are these ALK-independent resistance mechanisms. And so we used what you may be familiar with, next generation sequencing, and we also have an internal fusion assay to really look for point mutations or new genes fusions that might be sending extra growth signals besides ALK to help the tumor grow. We also did a test called FISH to quantify the number of copies of a gene called MET. We had some clues based on the EGFR space that MET may be something relevant in the context of ALK as well. And then in pairing with our laboratory collaborators, specifically Dr. Hada's lab, we used our extensive library of patient-derived cells to conduct these cell lines to conduct these functional screens to try to identify relevant bypass pathways. And so our genetic analyses, so putting aside the cell line screens for a second, told us that after alectinib or another second generation ALK inhibitor,
Dr.
we saw
Jack, your sound is coming in and out. I'm not sure whether you don't have quite enough internet signal. Can you hear me, Dr. Jack? Well, we're not hearing you. What we might try to do, I don't know, would this work, Keith, is for you to play the slides and just have Dr. Dagogo-Jack on audio. Do you think that might help? Oh, okay. All right. So I don't have, if you can hear me, Dr. Dagogo-Jack, I don't know whether you can hear me or not. I think we've lost you. Okay. No. We might move on to the next presentation, Dr. Dagogo-Jack, and then you can try coming back in again in a few minutes. Maybe. All right. Okay. So we had one more presentation, I believe, from Dr. Bar-Peled. Can we get him on?
Yes, I can. Can you hear me?
Terrific. Great.
And I'm sorry ahead of time for the background noise, but I had to step outside while my son was sleeping or taking a nap. But I'll try and share this very quickly. All good or no?
Everything's okay, apparently.
All right, great. Well, I really would like to thank everyone, and especially the ALK-positive and LUNGevity Foundation for sponsoring our research. And I will echo what many of the other panel or researchers have said. It's been wonderful to work with you guys, to interact with the patients. And I know many of you are intimately, you know, John Iafredi very well. He wishes he could be here. But today I thought I would tell you about some of the exciting research that we're doing in a very collaborative project. And to echo what Dr. Dagogo-Jack said, we're taking a little bit of a different approach here. While we think that immunotherapy is quite important, we thought that one really important way to overcome ALK resistance was to target cysteines, as I'll talk about. I think I'll start with something that many of you are familiar about, that is that genomic studies, such as those identified by the Cancer Genome Atlas or functional genomic studies from the cancer dependency maps brought out of the road, have really given us a good idea of those genes that are intimately involved in cancer. And from these studies, we've identified over something like 400 cancer drivers, and we've used this information quite effectively to be able to develop new classes of drugs which target these cancer drivers, including ALK. And I'm sure that all of you are aware that since 2007, since the first identification of the ML4 ALK fusion oncogene, we've been able to effectively drug the kinase domain of ALK, and that obviously brings a lot of benefits to patients. However, I think it's also very fair to despite our ability to drug ALK, unfortunately, again, as many of you are intimately aware, resistance does play an important role and is a natural fact with this disease, unfortunately. And so the question that John and I had was, could we find another way to target ALK-driven non-small cell lung cancer? And what I'd like to tell you about today is that we think we have a new approach. And if there's one thing that you'll take away from my talk, hopefully, is that cysteines, a very important residue with proteins, play a critical function. So cysteines, as I mentioned, are incredibly important in protein function. They act as nucleophiles and enzyme active sites. They coordinate metals. They're post-translationally modified. They form protein-protein complexes. And all of these are just different descriptors to try to remind the audience that cysteines are key components of protein. Importantly, because of their unique nature, they can function as sort of a chemical handle for us to build drugs with. And there have been many recent examples where covalent inhibitors that target cysteines have been successfully deployed in the clinic. Most notably, osimertinib, which is an EGFR inhibitor and targets EGFR mutant lung cancer. So we asked, could we target cysteines to identify potential new approaches to target ALK-driven non-small cell lung cancers? And so let me take you through our drug discovery. The first type of thing that we do is, before we start any project, is we make a map of all the different druggable targets within a particular cell line, and we've now extended this to patient tumors, which is incredibly exciting. And this gives us a high coverage map of all the proteins we think we can go after. Following our identification of targets, we build a tailored small molecule screen, which will read out the activity of that particular target, which we think we can go after. And we screen our lab's library of over 5,000 covalent molecules in order to identify a next generation drug. And from there, in our own lab, and also in collaboration both with colleagues as well as industrial partners, we do medicinal chemistry to take our hit compounds and make them into drugs. And I should mention, this is very much an iterative process, because as we develop more and more important leads, we always go back to the proteomics, not only to determine, but to determine off-target engagement, because we want to minimize the number of off-targets as we build new drugs. So I'm very excited to share with you the first ALK-positive non-small cell lung cancer drug map. We've never presented this in the past. This map shown over here is exactly like you would have a regular type of map. You know, instead of directing you towards your house, it directs us towards the target, the proteins that we think are going to be very, very druggable within ALK-positive lung cancers. And so each one of these particular each one of these little mountains, so to speak, is a protein target that we think is highly druggable. And we got very, very excited by the fact that we identified one of these mountains, one of these druggable targets as EML4. And the reason for this, if you'll recall my first slide, is that EML4 is the most prominent fusion within ALK-driven lung, the fusion partner. In addition to EML4, we identified multiple downstream components of the EML4 ALK pathway, which contain druggable cysteines. And so what this gives us is a wide variety of different approaches that we can go in that both target the main driver here, being EML4 ALK, and potential bypass pathways as well. So we knew that EML4 and ALK was druggable or contained a barely druggable cysteine. Now we have to identify a drug for it, or a small molecule inhibitor. And so we have a library of over 5,000 compounds. And instead of testing all of those compounds individually, which would take a very, very long time, what we decided to do in a sort of innovative approach was to pool the libraries together, pool the compounds together. So instead of testing one compound at a time, we tested 10 at a time. We called this our multiplex library. And to each of the then we took our favorite ALK fusion positive, added our multiplex library, and then we read out the activity of the 10 different small molecule inhibitors using a immunoblotting assay, which will read out many different parameters, including phospho-ALK, phospho-AKT, and phospho-ERK activity. If we identified a hit from this primary screen, we would then want to deconvolute that particular, this particular hit. And so now we have the 10 compounds, and we would re-assay it in the same type of assay and be able to identify the particular winner. All right, so how did this actual experiment look like? So here I'm showing you 10 different multiplex pools. Again, each corresponds to 10 different compounds. So in total, in one Western blot assay, or immunoblot assay, we're testing 100 different compounds. You can see here lorlatinib completely turned off ALK signaling. But very excitingly, many of these other multiplex pools also turn off ALK signaling. In particular, one that we really liked was this C9 compound. Don't worry about the name. And so we took that and we advanced it. Right now, we did the expansion screen where we test each of the individual compounds that go into this multiplex pool. And again, we identified one compound in particular, this 1C9 compound, which potently turned off ALK signaling. And as you can see in a dose-dependent manner, it turned off ALK signaling with a predicted IC50 of around 5 micromolar, which me as a chemist is very exciting, because to get such a potent inhibitor right off of a screening deck suggested that we were going to make a good inroad. I think something else that John and I are very excited about are the fact that this hit compound that we've identified, 1C9, not only engages wild type ALK, but engages all of the different or many of the different resistance mutants within the ALK kinase domain. And this is important because we're not targeting the ALK kinase domain, rather we're targeting EML4, which is right before. So as you can see in the Western blot here, wherever there's a positive sign, you can see close to a disappearance of the ALK signal. So to conclude, we think that our hit compound is a very promising hit with an exciting mechanism of action, which I'd be delighted to talk about. We're proceeding right now with the lead optimization and structure activity relationship to make this hit, to turn this hit into a lead compound and then be able to make it into a drug. There are many, many different steps, such as designing a large number of derivatives, determining protein-protein structure interactions, establishing whether this inhibitor can disrupt the activation of ALK-positive lines. And we're also in the process of turning this inhibitor into a degrader through PROTAC. So let me come to the most important slides, the acknowledgements. So this work would not have been done without John and his team, and in particular, very, very talented postdoc, Diane Yang. And the many members of our team also took part, especially with running the proteomics samples and the screen. And again, I would just like to thank the ALK-positive community as well as the longevity community for providing us really with this critical seed funding, which allowed this project to take off and to get to a very exciting step.
Yeah. Thanks very much, Dr. Bar-Peled, and also to your collaborators, Dr. Iafredi. I think it's really important to call out that we saw a new drug map. First time it's been displayed publicly, and it's a very tangible reminder that the work and the donations and the research that we're sponsoring is really driving best in class new innovations, and we have the privilege of being the first to see it. So we'll move on now to a pre-recorded presentation by Dr. King, if we can bring that up.
We have with us here Dr. Jennifer King from the GO-2 Foundation. GO-2 Foundation does lots of amazing work that Dr. King will tell us about. And also they helped us with selecting the awards for Dr. Trever Bivona and Dr. Christine Lovely in 2019. Thank you very much for joining us today, Jennifer, and I'll hand it over to you.
Thanks, Colin. As Colin said, I'm Jennifer King from GO-2 Foundation for Lung Cancer. I'm the Chief Scientific Officer. GO-2 Foundation is a nonprofit patient advocacy organization who works to transform lung cancer survivorship across the entire continuum of care. We have patient support programs, advocacy. We work with a network of centers of excellence that are different hospitals across the country, and of course, science and research programs. In 2019, we worked with ALK-positive to award research awards that would really look at how to turn ALK-positive lung cancer into a chronic or curable condition. We solicited a number of different proposals from different investigators, and in the end, 2 rose to the top, focused on understanding resistance to targeted therapies in the ALK-positive lung cancer space. The 2 investigators, Dr. Lovely and Dr. Bivona, had excellent proposals, and the review committees from GO-2 and ALK-positive worked with them to combine this into one coordinated proposal to really understand drug resistance in ALK-positive lung cancer. So I'm pleased to introduce Dr. Christine Lovely and Dr. Trever Bivona, and I hope everyone has a great meeting.
Thank you very much for that, Jennifer, and I'll hand it over to Dr. Bivona and Dr. Lovely. Thank you. Dr. Lovely?
I am, but I thought Trevor's presentation was going to go first.
Okay, sure, we can run that first then. So we've got a pre-recorded interview with Dr. Bivona, and then we'll have Dr. Lovely come on after that. Are you ready for that, Keith? So it's my great pleasure to have with me today Dr. Trever Bivona. Unfortunately, he's not able to attend on the day of the summit. So Trever is with me here a couple of days beforehand. And Dr. Trever Bivona is a board certified medical oncologist with a PhD in cell and molecular biology. He maintains an active academic clinical practice and clinical research program, while also leading a basic and translational research laboratory focused on cancer genetics and precision medicine. And I will add here that Dr. Bivona's lab is one of the best known in the world. A major research interest is enhancing the understanding of the molecular basis of targeted cancer therapy response and resistance in lung cancer and other solid cancers. He leads a multidisciplinary team of investigators in laboratory-based patient-focused investigation and is a principal investigator on clinical trials, enabling a bench-to-bedside research program. The overall goal of these efforts is to improve survival in molecular subclasses of cancer patients, such as ALK, through novel, effective precision medicine approaches. He's going to make a presentation about the research that he is doing that was funded through GO-2 Foundation by ALK-positive and how he expects that it could potentially impact our treatments. If you have any questions for Dr. Bivona, please email me at medical@alkpositive.org afterwards, and I will pass on the appropriate questions to him to provide answers. Dr. Bivona, thank you very much for joining us today.
Hi, Colin. Thank you very much for the kind introduction. And I want to first thank you for the opportunity to present, thank ALK-positive, and thank GO-2 for the generous support over the years. And again, my apologies for not being there on the day. And I'm certainly happy to answer questions or take comments from you, from the audience later. So I'm going to go ahead and share my screen. And then I have a few slides to show. Okay. Are you able to see?
Absolutely.
Terrific. Well, the title of my talk today is rather general. This is going to reflect the work that we've done in collaboration, with Christine over the years, and been wonderful collaborations over many years with Christine, even predating this work that we're going to describe today. And this is about new therapeutic strategies to enhance response to ALK-targeted therapy in ALK-positive patients. So the major focus of our work is to understand the evolution of resistance to inhibitors against molecular targets such as ALK, and that's what we're focused on here. And so this depicts, a lung cancer here before therapy and then after therapy. And while ALK inhibitors are certainly very effective in many patients, and in some cases very long periods of time, there are situations where resistance does develop. And that comes in the form of acquired resistance on the right of this slide here. And that's where patients are often resequenced, the tumors are resampled, or liquid biopsies are performed, and they're used to assign a different therapy or switch in therapy that might be more effective and able to address that resistance. One area of focus in our project has been in this middle component here, incomplete response or residual disease. And this is fairly typical for many patients, particularly with metastatic disease, where we often don't eradicate the entire tumor and cancer cells are left behind. The mechanisms underlying this persistence are relatively less understood, particularly in comparison to later acquired resistance. And so this was the major focus of the project here. Many years ago, we established a pipeline to deeply characterize these tumors before therapy and on therapy using a variety of approaches, one of which is single-cell RNA sequencing. This allows us to sequence the RNA of tumor cells and also microenvironment cells and understand the molecular programs that are going on at these different treatment states. And so one of the results that arose out of these studies many years ago was a particular pathway that seemed to be particularly enriched at that residual disease time point in the cancer cells, and that is the Hippo pathway, which involves a terminal effector called YAP, the protein YAP. This is a transcriptional co-regulatory protein that regulates the expression of many genes in cells. And we found this to be the signature of YAP activation and YAP itself to be enriched at this residual disease time point compared to the other treatment states, acquired resistance or the treatment naive states. These are some of the characteristic genes in this pathway. And we found also that this protein YAP, which is a nuclear regulatory protein, a transcription factor, a transcriptional co-regulator rather, is actually enriched in the nuclei of cancer cells in these patient biopsies, particularly at the residual disease time point here. And so this led to the hypothesis that perhaps this pathway, which is a known oncogenic pathway in many cancer types, might be supporting that residual disease state, meaning that the cancer cells are able to resist a complete eradication with ALK inhibitors. I should mention that we also found this to be more general and extended even beyond ALK to EGFR cancers as well, treated with osimertinib. So an example, we're studying, one type of lung cancer can actually help us understand another type of lung cancer and hopefully benefit patients as well. I'll go through a few data slides relatively quickly, and I've highlighted in red boxes the ALK-focused work as opposed to the EGFR work here. Again, similar findings. We found that this protein YAP is activated in these cancer cells by a variety of mechanisms. One Western blot here, another transcriptional analysis here on the right. And in fact, we found that silencing YAP here by RNAi-mediated approaches, so suppressing the expression of YAP, actually could address these drug-tolerant persister cells, which are an analog in these patient-derived models of the clinical residual disease samples. And that's just shown here on the lower left, where the cells are resisting therapy in the presence of YAP. And if we eliminate YAP using different approaches here, we can then sensitize these cells to the ALK inhibitor. We also found this to be upregulated in more patient-proximate models, such as patient-derived organoids and xenografts here, PDOs and PDXs. And again, here are some of the ALK-focused work down on the right here. These are PDX models of EML4 ALK-driven cancers, where we can generate that residual disease state in the mice by treating with alectinib here, in this case, an ALK inhibitor. And we find enrichment now of the YAP signature again at that time point. And so this leads us to the therapeutic hypothesis that perhaps targeting this survival pathway, this YAP survival pathway at the residual disease time point might actually enhance response to ALK inhibitors in mice and hopefully in patients in the near term. And in fact, it's quite fortuitous that there are inhibitors of YAP that are in the clinic now in phase 1, although not in lung cancer yet. And so we've begun studies with one of them from Ibiayi Therapeutics. VT104 is the preclinical compound. And we have initial data that I won't share with you today for sake of time, showing that, in fact, using this YAP inhibitor can enhance response to an ALK inhibitor. We've tested alectinib to date, and we want to test other ALK inhibitors as well in the future. And so the other aspect of this that is important to note, and I don't have time to discuss, that the YAP activation that we showed actually occurs through upstream proteins and pathways, such as another drug target for which there's a clinical drug now called FAK, or focal adhesion kinase. This protein actually helps promote YAP activation in this persister cell or residual disease state. And so we've begun to collaborate with Veristem, who has a first-in-class FAK inhibitor, VS4718, to test a parallel therapeutic hypothesis that another way to attack this problem might be to inhibit FAK in combination with an ALK inhibitor such as alectinib or lorlatinib or others. So with that, I'll end with that story. Just mention in 30 seconds one other area that's that we're beginning to collaborate with many labs on is our recent discovery that these ALK fusion proteins actually form a unique structure in cells, and that is they form cytoplasmic puncta, these cytoplasmic protein granules. And this is work that was quite serendipitous, but has led us to a model where in a non-conventional or unconventional manner, these ALK fusion proteins actually form their own subcellular structure that doesn't involve lipid membranes like other oncogenic drivers in lung cancer, such as EGFR and others. And that what's most important is that this structure, the formation of this structure, is actually essential for the oncogenic signaling downstream. So in order to promote cancer cell proliferation, this structure needs to form. And so we've begun to test a therapeutic hypothesis that perhaps physically disrupting this structure might actually be another way to attack ALK fusion positive lung cancer that could complement ALK TKIs and other complementary therapies as well. And so with that, I'll just end and thank obviously Christine for the wonderful collaborations over the years. A very talented postdoc in my lab, Francisco Herderk, who led the work on YAP in the role of ALK inhibitor resistance, and the granule work was led by Osman Tapuli. And again, thank you so much to ALK-positive and GO-2. And again, would love to entertain any questions going forward. Thank you so much.
Terrific. That was a great presentation. Sorry we had to have it so short. So Dr. Bivona has given us a glimpse of what he's working on at the moment. And we certainly hope that those drugs through Vivace and Veristem get into clinical trial before very long. And we'll be hearing more about that before too long, I expect. And then Dr. Bivona has also given us a glimpse into his future research about granules. He's very, very, very excited about that, I know indeed. So thanks again for joining us today. And we look forward to seeing your research continue. And obviously the goal is to get to clinical trials and get something that makes better outcomes for us. Thank you so much.
Thank you. I agree, and wish you all the best.
Thank you. So you've probably all heard the saying, my overnight success took 10 years to get. And it's like that with research. So you see the headline, new discovery, but it takes a lot of background work like this to get there. And so it's coming along. He's in conversations with companies, getting the drugs to get to the next level. We've got Dr. Lovely. We're going to be skipping the international research. They're pre-recorded, and we just don't have time for those with some of the other presentations having gone over. They will be uploaded to all the ALK Summit recordings, so you will be able to view those once they all get loaded onto the website. So Dr. Lovely, thank you so much for joining us. And it's wonderful to see you. I wished I had you here, but that's the next best thing. And Dr. Lovely is going to talk about her collaborative project through the GO-2 Foundation with Dr. Bivona. Thank you.
Hi, everyone. Thank you so much for hanging in there with us. Last, but hopefully not least, and just for a quick intro, I'm Christine Lovely. I'm a physician scientist at Vanderbilt University in Nashville, Tennessee. I had the best time at this meeting in 2019 when it was in Atlanta. I wish I could be there today. I'm going to jump right into my slides. Okay, hopefully you can all see this. So again, my name is Christine Lovely. I think this picture is very appropriate since you all are in Denver and it's just a beautiful location with beautiful mountain settings. My research team, so I'm truly a physician scientist. I have a clinical practice and I have a laboratory. We really think about the clinic and the research lab, the experimental lab as our great research opportunities, taking clinical observations to the research lab, questions that we find in the clinic, and then ultimately bringing those questions and that science back to the clinic to help patients. And we love having patients visit the lab. We had a great visit with the ALK-positive group in February 2020, right before the pandemic started. We'd love to host anyone else if they're ever in Nashville to come and see the lab and actually tour what a research lab looks like, because most people have never been in a research lab before. So I'm going to, my presentation may be a little bit different than the previous ones, and I apologize for that. But I'm going to summarize a couple of different projects very briefly and high level. So broadly speaking, our goal is to expand the reach of precision medicine for patients with ALK-positive lung cancer. And I chose this slide because it's baseball season, and this guy is expanding his reach to catch the ball. And what we want to do is that same principle, extend our reach, really make research practices better for patients with ALK-positive lung cancer. And how are we working to achieve this goal? Well, 4 big things. So first, like many of the other speakers you've heard about, we want to develop better drugs and better combination therapies for ALK-positive lung cancer. The second thing we want to do is really capitalize on available data sets. So a lot of the research that's very provocative and very helpful actually just looks at data that we already have in hand. And so I think it's important that we use everything in our toolbox to study ALK-positive lung cancer. We want to extend what we know about ALK-positive lung cancer to other tumor types. And finally, and most importantly, and I'm going to start with this message, is you're hearing about a lot of great research today. We also have to, as we drive research forward, make sure that we are delivering on what we already have. So there are still too many patients in the world whose tumors are not tested for ALK. And so we need to continue to push the research, but we also need to continue to push the implementation. We want to be able to actually deliver on and give patients what we have as our best right now while we continue to develop even better therapies. So my first goal is to develop better drugs and better combination therapies to prolong response to anti-ALK drugs and to overcome ALK resistance. How are we doing that in my lab? Well, I'm going to summarize a bunch of data in one slide, but we found this protein. It's an anti-cell death protein called clusterin, and it's a potential new target to help overcome resistance to anti-ALK therapies. So what is clusterin? Big picture, clusterin is a protein inside the cells, and it is made by cells to protect the cells. It has a protective function of the cells. It actually may help cells escape from the effects of drugs that promote cell death. So this is like a way that the cells can say, okay, the drug is present, but we still want to live in the presence of the drug. So we're going to, express this protein called clusterin. That said, the exact way clusterin behaves in a cell is still under a lot of debate, as is a lot of the science you're hearing about today. So, I want to give that important caveat. You've seen this kind of data before, but big picture, what we did was we took a bunch of ALK resistant cells, so ALK-positive lung cancer cells that are resistant to numerous different ALK inhibitors. And don't worry about the labeling too much, just the big picture resistant cells. This blue line is the ALK-positive cells that are sensitive to the ALK drugs. This red line here is the ALK-positive cells that are resistant to all the ALK TKIs, all the ALK drugs that we have. And what I want you to see here is very simple, is that if you look at this black blob, this is clusterin. So in the resistant cells, there's a lot of clusterin, but in the drug-sensitive cells, there's essentially no clusterin. And what we found that if we get rid of clusterin, we actually make the resistant cells respond better to ALK drugs. And so let me walk you through this for a minute. So this experiment here, it's what we call Western blot. You've seen them before, but don't worry about that too much. These are cells, an ST1, a cell line that is resistant to lorlatinib. The green line is if we look at here, these cells have a ton of clusterin. So the resistant cells have a ton of clusterin. The orange line is if we use a method to actually get rid of clusterin in the cells. And so this is just showing you that we can get rid of clusterin. If we look over here, this is just counting cell number. And again, the green bar here, the green arrow, is the cells that are just happily growing in lorlatinib. They're resistant to lorlatinib. They're happy to grow in lorlatinib, no problem. The orange lines are when we actually get rid of clusterin. Now the cells actually start to die when we put clusterin, when we get rid of the clusterin protein. And hopefully you can see the difference here. These are cells in the green bar, happily growing in lorlatinib. When we get rid of clusterin, the cells are still growing, they're still in lorlatinib, but now they're actually dying. And so clusterin helps those cells to die because it has a protective effect. So why is this important and interesting? Clusterin has a potential protective effect for cells, and we want to get rid of this protection. We don't want cells to live in the presence of these ALK drugs. We want them to die, and we want to get rid of all of the mechanisms they have to survive. I would say it's important because we need new targets to combat with anti-ALK drugs to make anti-ALK drugs work better and longer. This is a huge goal for many of the research projects you heard today. And there actually is a clusterin inhibitor already in early phase clinical trial, not for ALK yet, because we're still gaining a lot of information on the dosing and how safe it is in patients, but stay tuned. This will be a continued big push for us. So pivoting to our second goal, which is to learn more about resistance to ALK TKI therapy using available large data sets. And so let me take a step back for a second. Something you haven't really heard about yet during this seminar is the 2 types of lung cancer. The majority of lung cancer we talk about is non-small cell lung cancer, but there's this other type called small cell lung cancer. It's about 15% of all lung cancers. And small cell lung cancer is different. It's more aggressive. We have less therapies, and it is just a different beast than non-small cell lung cancer. So a project that I've been working on, and it's been submitted for publication, we worked with a genotyping company called Foundation Medicine, and we looked at their entire cohort of patients with small cell lung cancer. And their cohort was 3,600 patients. And just for frame of reference, the largest study published in small cell lung cancer to date is about 100 patients. And what we found is actually there are multiple cases in this data set where a patient with ALK-positive non-small cell lung cancer had two samples, where one sample was ALK-positive non-small cell lung cancer, and then the second sample was ALK-positive small cell lung cancer. And so this is one example of a patient, a 48-year-old gentleman who had ALK-positive non-small cell lung cancer. And 722 days later, after the patient became resistant to the drug, they had a repeat biopsy, and now we see small cell lung cancer with a different variety of mutations. The ALK fusion is still there, but now there's these other mutations, and what we observe is small cell. So why is this interesting and important? Well, small cell lung cancer is a more aggressive type of lung cancer that is treated differently than non-small cell lung cancer. And it's important to note that repeat biopsy is not required at the time of tumor regrowth on anti-ALK therapies right now. So that's not absolutely required. But if you want to detect a change from non-small cell lung cancer to small cell lung cancer, you need a biopsy. And so this data really suggests that we should be biopsying patients with ALK-positive lung cancer at the time of progression to really make sure that if we find small cell lung cancer, we're treating it appropriately because it has a totally different treatment regimen than non-small cell lung cancer. Last goal, to extend knowledge about ALK to other tumor types. This will be very brief, I promise. As you all probably know, we're talking about largely about ALK-positive lung cancer, but ALK is found in many other tumor types as well, and particularly a type of sarcoma called IMT, which has a big fancy name, inflammatory myofibroblastic tumor. Well, I was lucky to work with a pathologist named Cheryl Coffin. Here she is with her dog, Anna. And if you know me, you know I love dogs. We compiled the largest group of this IMT tumors ever studied to date. And we did this special sequencing. And what we found, interestingly, was that more than 50% of these tumors, and these tumors I failed to mention, it's a rare tumor and it's predominantly in children. More than 50% of them had ALK fusions, and some of the same ALK fusions we find in lung cancer, such as EML4-ALK. And let me show you one case. This is a patient of my colleague, Scott Bornstein. This is a 2-year-old little girl who had an ALK-positive IMT, and she has this big tumor. Hopefully you can see my mouse in her chest. And she went on an ALK drug, and that tumor basically went away. So why is this important? Well, until July 14th, just 2 weeks ago, ALK therapies were not available for patients with IMT, but in compilation with FDA, Pfizer, and a bunch of investigators, finally now crizotinib is available for patients with ALK-positive inflammatory myofibroblastic tumor. And I think this is incredibly important and interesting. Why? We know a lot about anti-ALK drugs in lung cancer, but less so in other ALK-positive tumors. We want to be able to share knowledge about ALK as a driver mutation across different tumor types, because we want to be able to offer patients with ALK-positive IMT or ALK-positive other tumors the option for these FDA-approved therapy options, the ones that we're giving to lung cancer. And I think it's very important as we as a cancer community actually share our knowledge. I will end by saying thank you. This is another picture of the ALK-positive group visiting my lab right before the pandemic, and close with some of my favorite pictures of some of my favorite people. As I'm sure you recognize many of them. I want to really give a shout out to Gina, who always said, see the stars, and end with my favorite expression, which is hope. Have only positive expectations. We can do this together as a community. Thank you all so much.
Thank you so much, Dr. Lovely. It was a great presentation.